strongly suggesting that the activity when you're looking at basically nothing, a grey screen, or even your eyes closed, is, at least in animals, is very, very similar to the activity when their eyes are open, which suggests that most of the activity in looking at stuff, so most of the activity is actually not then encoding stuff that's coming in, but also is doing some ongoing processing. And the ongoing processing, the best guess is what it's doing is just simply predicting the features that are going to be there. Each layer is predicting what the layer below it should be sending it. So V2 should be predicting the elementary edges that V1 should be seeing from the fact that it's predicting these lines. V4 should be predicting the lines and the corners that V2 should be sending it because it should be seeing the outlines of these objects, and so on up so down the hierarchy. What it actually is then just the difference between what's coming down and what's coming up from the retina.
Yes.
So what's being passed forward is the thing that needs to be tweaked in the predictions.
Welcome to Stimulating Brains.
So hello there and welcome back to Stimulating Brains episode number 13. This time together with Mark Humphries, who is the chair in computational neuroscience of the Faculty of Science at the School of Psychology, Nottingham, UK. Mark has just published a book called The Spike, an epic journey through the brain in 2.1 seconds with Princeton University Press, which is a highly recommended read. It's really entertaining. It's also easy to read, I'd say, and still very informing. And beyond talking about that book, of course, Mark and I talk about a new paper he just published on 2 principles of dimensionality reduction in the brain. And going back a bit further in time, also about his PhD work together with Kevin Gurney, who, of course, together with Prescott and Redgrave, developed one of the most influential computational Basal Ganglia models in 2001. We also have a slight detour in there about the question, do you even need the cortex in the brain? And yeah, it was a great privilege to be able to pick Mark's brain. I think his work is really excellent and outstanding, and I'm pretty sure you're going to like the conversation we had. So thanks for tuning in once more. Stimulating Brains episode 13. Have fun. Great. So thank you so much once more, Mark, for taking the time to do this. I will have introduced you more formally by now, but to break the ice, I always start with the question, what do you do when you're not involved in research? So in your private time?
That's a really good question. Yeah. In what? So what private time I have away from doing research and obviously family with young kids. So in that smidgen of time, it gets split mostly between sort of traditional academic pursuits of reading a hell of a lot of stuff that is not anything to do with what I research. So I just love writing and all sorts. So I read all novels of all stripes, and non-fiction of all kinds of history and science and so on. Particularly love evolution.
Oh, great.
Stuff. So I'm not quite sure why I didn't end up being an evolutionary theorist, because I spent my entire teenage years reading Stephen Jay Gould and Richard Dawkins books and thinking, wow, theoretical evolution is great. But ended up being a computational neuroscientist instead.
Sure.
And the other pursuit mostly of music. So a lot of play guitar.
Oh, great.
Historically in a bunch of fairly mediocre bands of various kinds, but these days mostly just for fun.
In fact, we share that. I also played in mediocre bands all my younger years. Yeah, has gotten much less. Do you have any good recommendations for evolutionary books to read for us?
That's a good question. I have to say, I recently reread Richard Dawkins' Blind Watchmaker, and it's still amazing.
It's great. Yeah.
Irrespective of his sort of social media persona, which is a little bit
true.
contentious these days, he remains a master of writing for a lay audience. Both in terms of it's just his style, let alone its content. He's a masterful writer. And the Blind Watchmaker, that's probably his best book. I mean, The Selfish Gene is the most, obviously, I know it very well. It's the most famous. It's a beautifully argued piece, but it's a little bit too, it's a little bit too, I don't know, sort of committed to the argument. It goes in really strong.
Yeah.
But as a blind watchmaker is much more nuanced. It's takes you much more into the highways and byways of the arguments about sort of the deep thinking of evolutionary theory about how genes are the element of evolution and what that means in terms of both phenotype and in terms of what we would how things came to be evolved. He spends a lot of time as a blind watchmaker, of course, talking about how complex things appeared. Given this, we think of this the unit of hereditary and of evolution is the gene. How on earth did you end up with eyeballs and stuff? So that he spends a lot of time on that. The sort of big evolution questions.
Yeah. Great. I also like that. I don't know the English title with the rainbow, something rainbow. I think it's the positive opposite of the selfish gene that he wrote in response to making a more positive claim and things.
Unweaving the rainbow, I think.
Unweaving the rainbows. Rainbow. Yeah, that I really liked as well. Anyways, we're not here to talk about Richard Dawkins, unfortunately, but about neuroscience, I guess. And you've done exceptional work in the field of computational neuroscience, maybe even theoretical neuroscience. I don't know if you would identify with that a bit. Would you mind telling us a bit about your broad take at research and what your lab's focus is? Is that even possible?
It's a good question to ask of any computational neuroscientist, because so many of us don't have a focus as much as a sort of bunch of things that we like to do and find problems that we like to apply them to. So for my lab, so the bulk of what my lab does is focus generally on the problem of how populations of neurons do what they do together. So how when you have a whole bunch of them active at the same time, that creates more than the sum of their individual capacities. Particularly, we're interested in cases like where we can decode and understand things from groups of neurons that are invisible when you look at them individually. So that's one of our things. Yeah, exactly. So we want to take sort of computational views of how groups of neurons do what they do, and we do that in the sense of analyzing lots of data as opposed to building models of neural computation these days. So I often tell people we're a neural data lab. So we're computational neuroscientists, but we take experimental data to answer questions about how populations of neurons encode things or what we can decode from them, and the dynamics they have when they are encoding these things. And because we're interested in that as a general problem, we don't focus on a specific brain region or a specific species. So we have worked on things ranging from sort of encoding of the present and the past in prefrontal cortex in rats to the encoding of the galloping escape movement in the sea slug aplysia in its motor centers in the pedal ganglion, so-called.
And you recently looked at 10,000 neurons simultaneously recorded from the Carsten Stringer data as well, right? So it's also a lot about high volume data these days, or is that true?
Yeah, so we have looked at data sets on that scale from some data from Carol Svobod's lab, where they've a slightly earlier data set than the Stringer data set, where they recorded from pretty much every single neuron in a single barrel of the barrel cortex of a mouse. Okay. So simultaneously, they recorded a maximum of about sort of 1,700 simultaneously.
Okay.
But when they stitched the recordings together, they meant basically they recorded from every neuron in layers 2, 3, and 4 of the barrel cortex, which is a pretty exhaustive survey of a tiny chunk of tissue. But in this case of this particular mouse, it's the barrel corresponding to the only whisker that it had intact on its face. So all sensory information from the whisker was being passed through these neurons. So whatever those neurons knew, the rest of the brain knew, which is great sort of bottleneck to ask all sorts of key questions about.
Great. Yeah. That sounds good. So, and we'll dive into that more, but maybe to go back a bit in time, you recently published a retrospective look-back paper on the Gurney-Prescott-Redgrave model of the basal ganglia from 2001, together with Kevin Gurney. And in it, you wrote, On the first day of my PhD in October 1998, I was handed a thick 51-page report held between covers of pale blue card on which were printed the ominous words, analysis and simulation of a model of the basal ganglia. What was that about in the beginning of your PhD?
So, yes. So the bit I missed off the answer to your question about what my lab does is I am historically a model of the basal ganglia, so I spent a lot of time building computational models of that chunk of the brain. And that report I was handed at the first day of my PhD was, so my supervisor, Kevin Gurney, had literally, as I started, just finished what had taken him, I think, 3-4 years of building this model of the basal ganglia, which realized this theory that the basal ganglia collectively do some form of selection between competing inputs, and in particular that they solve the problem of action selection, of the problem of choosing what to do next. So this report then was what ended up being 2 papers in biological cybernetics published together, because it was 51 pages long, so it was quite dense. The first half was really sort of a review of all the anatomy of the basal ganglia from a sort of a theorist's eye, going, if we think the basal ganglia does something to do with action, and we look at its anatomy, the wiring inside it, all the inputs it gets, and the way it's set up, and the scaling of the neurons and so on. Sort of how do we interpret that anatomy in this context of action selection? So that's the first sort of paper, was saying, okay, we start from all this information and we distill it down into a sort of a wiring diagram that says this wiring diagram looks as though it should be able to do some form of action selection. The question is, does it? So that's the whole point of being a modeler, is that you take that wiring diagram, you build a model of it, and then you show, does it or doesn't it? And what exciting things pop up along the way that you weren't expecting. So the second half of the report is this model in a simple sort of population level dynamic model, which is then analyzed for all of its stability and selection properties, and then simulated towards the end. So it's particularly the page after page after page of the sort of the analysis stuff that was at the start of my PhD completely lost me.
I can imagine, yeah.
Yes.
So another famous model of the basal ganglia is, of course, the one by Garrett Alexander, Malum DeLong, and Peter Strick from around 1986, or I think more frequently now people refer to it as the Alban DeLong model from 1990. As younger students of the basal ganglia weren't there at the time, so it's sometimes hard in retrospect to even understand, what each model added. So could you maybe help us disentangle the two? What was new in the Gurney-Prescott-Redgrave model, and how do they differ, or did it build upon that?
Yeah, so the original sort of direct-indirect pathway model, as you say, has this sort of tangled history of a few papers. And if you go back and read them, there's no one paper in which it clearly says this model is.
Yeah.
It sort of emerged out of those sort of collection of papers. There was Mahon DeLong's sort of TINS paper and the Crutchell Alexander one in the same issue, which they kind of 1990 sort of formalized this. Obviously, they were just wiring diagrams, and they were also quite simplified wiring diagrams. They had knowingly omitted some stuff just to sort of get a view of the world that was you could think through in your head.
Yeah.
And the key aspect they were emphasizing in that model was obviously that you have 2 pathways that go from the striatum to the output nuclei of the basal ganglia.
Yeah.
So one population of the striatum sends this direct projection to the output nuclei. So you got direct projection neurons to the output nuclei. And the other one goes via some intermediary nuclei. Okay. And the way they originally drew the indirect pathway, they have a sort of 3 synapses, so 2 regions interposed. So they have the population of the striatum projecting to the globus pallidus projecting to the subthymic nucleus projecting to the reticulata. So you have this indirect pathway meeting up with a direct pathway. And so one question you want to, when we build models, is just to ask the question, well, you say that works, but does it?
Sure.
And the answer is, when you build a model of that actual pathway, it doesn't do what they think it does. One reason for that being that the entire control of the indirect pathway relies on if you just add up the signs in your head, you have this inhibition from striatum inhibiting the globus pallidus, which itself is inhibitory. So you turn up the striatum, you turn down the globus pallidus, and you have this net excitatory effect.
Sure.
Except that actually, you don't have to turn striatum up that much to turn off the globus pallidus. And at that point, the indirect pathway has no extra effect whatsoever on the output.
So the minus times minus doesn't work anymore if you turn it off completely.
Exactly, it's just gone. So there's no longer any competition between the two pathways. And what also misses out is, obviously, there's a feedback from the subthalamic nucleus to the globus pallidus. So one of the big things investigated in the Gurney model was this feedback loop.
Yeah.
And the other sort of key thing in the Gurney model was also the indirect direct pathway model is just literally just two pathways. It's just literally one set of arrows and another set of arrows. It doesn't address the question of how do you represent multiple things in this structure.
Sure.
And as soon as you have more than one thing you need to represent, you need to decide whether you're going to how you're going to make those things meet up again. Whether they're going to follow exactly those two pathways and just meet. So if you have action 1 in the direct and indirect pathway, they meet as action 1 of the output. Action 2 meets as action 2 of the output, or some different combination. And again, so part of the thing with the Gurney model was an exploration of, we actually expect that the subthalamic nucleus output doesn't go to a particular action. It goes to all of them because its output is the axons into the output nuclei go everywhere, so they don't go to a specific topographic location. Yeah. So we expect them, no matter what the representation is, they just go over the whole lot. So there's two of the ways in which it is. Yeah.
And in that realm, the SDN would be really important, as I understand it, in that model, in the Gurney model, to do switching, right? Is that correct?
Yeah. So in the Gurney model, certainly the subthalamic nucleus, that diffuse output is key to its operation. In particular, that the ability to turn an already selected action back off, essentially by the fact that if you have new options arriving to choose between, the subthalamic nucleus activity should then grow, and that growth excitation should pull the activity of the output nuclei back up again, away from having shown some selection with disinhibition, and then the new inputs arriving should hopefully drive one of those outputs down again. Right. One of the things that was a little bit missing from that paper was a detailed analysis of that process. It just says, so this should happen, but obviously there's at least lots of scope of, well, what kind of balance or do you need of the inputs to be able to have enough inhibition to overcome the extra excitation and that kind of thing.
Sure.
So that's where simulation comes in to do that complete sort of scope that out.
Interesting. So, and I guess as far as I understood, the Gurney model then, of course, was really influential in the field of especially computational neuroscience. And one prominent example that I know of and that you also cite in that retrospective paper is the inclusion into the SPAWN model created by the team around Chris Elliott-Smith. What is SPAWN, and why was the GPR model more important in that model?
Yeah. So I'm going to slightly a vague answer here because it's been many years since I read the SPAWN paper in detail.
Sure.
But obviously the SPAWN paper it was 2016, I think, or 2015, something it must have been earlier. Yeah. So it was a science paper on this 1 million plus massive spiking neuron model, in which the goal was have a single model which was plausibly brain-like, so it had spiking neurons connected together in a big network that had different plasticity rules arranged into different regions, one of which was a basal ganglia-like architecture.
Yeah.
That could learn and then perform a range of different cognitive tasks. So I think they tested on 8 sort of simulations of different tasks you'd give a human, and show that the model, without any sort of special reparameterization, could learn all of these tasks and perform them using the exact same architecture. So within that model, because obviously most of the tasks were about performing some kind of response, so some kind of choose left of classic human response task, left or right thing, for either kind of reaction time stuff or just decision making stuff. So you needed something to choose the responses. So the basal ganglia component, as I understood it, was the part which was central to all the as we designed it originally, the action selection part of the process. And the model they used was literally just a spiking rendition of the Gurney model, even more literal than the one that we built. We built one which we published in 2006, which had all sorts of extra stuff in it to be more sort of biophysically realistic and show that this dynamics action selection all works while you can still replicate a whole bunch of electrophysiological data, but the Elias Smith team literally took the Gurney model and replaced the populations with spiking neurons, and it just worked. What they added though was they added synaptic plasticity to the striatal inputs so that they could have it learn the responses while the rest of the spawn architecture was learning the structure of the task.
Sure.
So their model was, in that sense, a little bit more advanced. They had it learning away. So that was really cool. So in the core of this massive model, there was, as we predicted in the core of the brain, this group of nuclei sitting there, constantly learning the correct responses in different contexts, given the same architecture for every single different task.
It really is amazing. I mean, also that, as you just said, they literally used it and it worked. So that must feel great, I guess. Would you think the model is still correct? Or since you said 2006, you did some additions, now it's 2021. What do you think about the basal ganglia? Is that still the main role with action selection or?
That's good. So it's certainly so the model, that original Gurney model. So I finished a little retrospective with thinking about. Of course, over the many years, it has been tweaked and embellished in many, many, many ways. To the fact that even the last versions that Kevin has been working on are sort of quite extensive sort of ornamentation built on top of that original core. And there's a sense in which you have to ask, at what point is it no longer the original model? Is it now something of its own kind? So we see the original model. So we don't think is an accurate depiction of what we now know about basal ganglia anatomy. Hence why we have to embellish the model over and over many years. Sure. But of course, then obviously but that's true of any model. No matter how advanced the model is not all of biological reality. It remains that the original Gurney Prescott-Redgrave model is useful as a way of thinking about how the basal ganglia work, because it sets out an entire way of thinking, which we touched on earlier about the subthalamus nucleus role in switching, thinking about how it's wired up in order to do selection, still make predictions that are testable. So, because if it turns out you can do action switching without subthalamus nucleus in your brain at all, then clearly that's wrong. So that would be a good start. But in terms of sort of the broader basal ganglia theories, the action selection theory remains as one of the theories that people are entertaining about what basal ganglia do. Yeah. And particularly of late, thanks a lot to the work of Josh Studman in particular, and obviously Robert Turner and others who've done a lot of work on how basal ganglia activity encodes the kinematic variables of movement. They're setting up a strong case for it's not doing action selection. What it's doing is it's setting up the parameters for the action you're doing. And these two can look indistinguishable, because you still need the actions to be represented independently if you're going to control their kinematics. You still need to independently represent the movement of your arm from movement of your leg, because the kinematics is separate, so they are controlled separately. So it looks like you have separate control of these things. But nonetheless, they believe their evidence suggests that you have basal ganglia is there for this defining the kinematic parameters of movement, particularly how sort of vigorous you do the movement, as opposed to sort of de novo selection of the thing you're going to do next.
Interesting. Okay. Great. So, not yet completely solved, I guess. Of course. So you recently wrote a book called The Spike. I think an epic journey through the brain in 2.1 seconds. That was with Princeton University Press. It's an amazing book. It's a fascinating tour de force journey through the brain. And in it, we follow a single action potential from the retina via visual to parietal to presental and so on cortices. Can you tell us anything about eating cookies in an office environment? Yeah.
So the book is written from the perspective of obviously you, the reader, as you say, following spikes. Individual spike through the brain from the retina onwards through the early visual system, region of the cortex, even all the way down to the spine eventually. And I situate the reader in a fairly sort of everyday office environment in which they're faced with a everyday but sort of non-trivial choice, which is middle afternoon, you're feeling a little peckish, you're about to go into for a meeting, you want someone to pick you up, and you notice that on the desk in front of yours, there is the last cookie in the office cookie box. And the ethical dilemma is, you're really hungry, do you get to take the last cookie? I guess depending on your culture, that's either a really big ethical dilemma or a small one. So in Britain, that would be a huge ethical dilemma. Do you take the last cookie in the office cookie box? In other cultures, I imagine, not quite so. Anyway.
Interesting.
But the whole point of the setup of the story then was to give elements that obviously would feed into that thought process of first being able to describe just how your visual system is picking up all this information around you, how it's resolving the fact that there is a cookie and it's in a box and it's in a desk, and where it is and whether it's moving or not, and how far away it is from you, and how all that information gets processed. And that all gets happening in the first 150, 200 milliseconds after your retina falls on sees the cookie. And then there's the process of deciding whether you're going to take it or not, and then in that process, trying to remember where people are around you in the office, who's looking at you, where people are moving away from you or towards you, so sort of working memory stuff, and then deciding, so based on evidence around you, whether or not you're going to take this cookie. And having finally decided to take it, then the spikes needed to move your arm, and rather than do anything else, to reach for the cookie and take it and eat it to give away the ending. But the reason why this ends sort of 2 seconds is it's a rough estimate of the total processing time with some cogitation in between, or how long it's going to take you to see something and decide to do something about it.
Why 2.1 though? Is there a reason for the 0.1?
No, the reason for 0.1 was to make it more interesting. Yes,
I figured that. Yeah,
there was a specific yes. So obviously, we're looking at a period of time of, we know it's going to take longer than a sort of classic sort of a visual response loop, which could be just less than a second of seeing something to reaching for it, all that sort of decision making stuff in between. A couple of seconds is pretty typical for an everyday action to be enacted. So yeah.
Makes sense. So in the book, you explain a lot of things, and I really can recommend it. But this podcast is explicitly really focused about brain stimulation, and you also seemingly explain how deep brain stimulation works, but you keep it quite brief in the book. So it seems to have something to do with synaptic failure. What's that?
Yes. Yeah. So glad you picked up on that, because that's one of my favorite little bits of the book. But it had to be short because it's kind of like a little interesting detour for the reader, the general reader who's going to be doing a tour of the brain. I'll be wondering why in this chapter in Cortex am I reading about deep brain stimulation. But let's see, so synaptic failure is just the general idea that synapses are not reliable. And it's even I would wager most people who call them neuroscientists, which is a pretty broad church, right? If you go to the Society for Neuroscience annual meeting, there's 30,000 people who call themselves neuroscientists, many of whom would have never have thought about how synapses work. Because it's there's too little time in the day for everything, every scale. So synaptic failure then is the fact that when a spike reaches a synapse to a postsynaptic neuron, the release of the transmitter often doesn't happen. So textbooks were taught spike arrives at the synapse, the calcium floods into the synaptic, the boson, and that calcium influx forces the vesicle into the membrane, diffuses through the membrane, it releases the transmitter, it locks into the other side, blah, 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 blah. What actually happens is the spike turns up and nothing happens. And it happens, in some synapses, the failure rate is enormous. So there are recordings in hippocampus of failure rates of 90%. So only 1 in 10 of the spikes arriving at a particular synapse hippocampus will cause any effect on the neuron on the other side. And the average in studies in bits of cortex is about 75%, I think. Honestly, when this first discovered, and there's this widespread, also a lot of theorists were wondering what the hell it's for, because theorists love these sort of questions. And it was a nice theory took hold that really, what's great about failure is it's a sort of, from a dynamics perspective, is that it's a low pass filter. It means that if the spikes coming in are generated with a particular pattern, that pattern is lost to the postsynaptic neuron. It can't see it because there's so many spikes drop out of the pattern that the pattern is. So if the spikes were an oscillatory pattern, that oscillatory pattern would be filtered out. to some degree, because obviously the spikes that happen at the peak of this oscillation would be massively lost, whereas the ones at the trough. So it would really thin out the oscillation.
Yeah.
Which brings us neatly then to this theory of how deep brain stimulation works. Right? So the idea is that, obviously, in the Parkinsonian basal ganglia, a bunch of the structures, particularly the subthalamus nucleus and the globus pallidus, are oscillating really strongly. And that oscillatory system is entraining the output of the basal ganglia, so it's now sort of jamming the basal ganglia targets to not be unable to be response to their inputs, hence the sort of akinesia and bradykinesia of Parkinson's. And then what deep brain stimulation does when it's put into the subthalamus nucleus is that, because it's driving the subthalamus nucleus axons so intensely, so this lockstep 100 hertz, what actually it's doing is it's causing, because it's so intense, it's causing a massive failure rate at the end of the subthalamus nucleus axons. So it's filtering out the input they're actually sending to the pallidus, which removes the entrainment to the oscillation. Because it removes the entrainment to the oscillation, then it resets the system back into firing normally.
And that would be pathological noise, maybe the beta power signature that we describe, right, in SDN or GPI that's present there. And that's probably pathologic. We see that more in PD. It codes for symptoms and so on. And then with the 130 hertz stimulation, we tone that down by inducing synaptic failure, I guess, is the idea.
Yeah, exactly. So that the thing that we think of as, so the bursting of the sort of individual neuron bursting we see in the animal models of Parkinson's, where we assume is what the beta band in the LFP in humans is, and then that indeed, so that the 130 hertz stimulation is knocking that out by forcing the sort of the synaptic failure to filter out all this oscillation that's happening, so that you no longer entrain, particularly the GPI, no longer entrain it to the oscillations that are happening.
And DBS to the GPI would also work. Would that be doing the same thing on the GPI level, or?
So that's good question. So who knows? Because the theory was worked out. It was from John Rubin's team. I think working with Isar Bargad. So they put together bits of this story. So John Rubin's team already had a paper about how you use how synaptic failure can act as a sort of low pass filter. And because he's a basal ganglia researcher too, I imagined something in his brain went, I bet that will work for deep brain stimulation. They put together this nice little paper in Neurobiology of Disease, which showed that it works. But that's specifically for subthalamic nucleus DBS, where you can see it makes sense. And so it would be a separate modeling question of, does that actually happen for GPI? Because one of the questions with GPI DBS, which foxes a lot of basal ganglia researchers like me, is if the GPI is the output of the basal ganglia, we understand subthalamic nucleus DBS works because it restores the output structure to be able to do what it does. Yeah. But if you put the DBS stimulator in GPI and just entrain that to 130 hertz, and how does anything at the other end respond when you've entrained the output structure you thought you were returning to normal? So one guess would indeed be that what it's doing is it's simply removing GPI's influence completely from thalamus by having synaptic failure, perhaps, and then it's just restoring the ability of the rest of the brain to work rather than restoring basal ganglia function of any kind.
That would make sense because a lesion to GPI also works, right? So,
right.
It is. I guess I personally agree with you that there must be something at a oscillatory level that toning down pathological signals is key, and that is probably possible with different mechanisms. But really interesting. So. Maybe going back to the book and synaptic failure, you also talk about type 1 and type 2 dark neurons. What are those?
Yeah, so these are... You
can start with one if you want.
Yeah.
They're very different. Good question.
So the dark neurons are the general idea that the neurons in the brain are a lot less active than we think they are. So historically, we also recorded individual neurons in the brain by lowering thin slivers of metal, thin electrodes down into cortex. And also you're doing that blindly. So the only reason you know, like you have recorded from an individual neuron is because you can see the spikes on the oscilloscope or hear it on the lab speakers. And that's how it's been done since the 30s and the original sort of recordings. But that, of course, means that you can only record neurons that are firing spikes. So it looks like all neurons are firing spikes because the only neurons you're recording from are the ones firing spikes. When you start recording from neurons, just generally, just unbiasedly recording neurons, it turns out that many neurons aren't firing any spikes at all for much of the time that you're looking at them. And that's what we're calling the dark neurons, or at least the type 1 dark neurons. So generally when I've written proofs about saying dark neurons, we mean this set who are barely active. And obviously they raise a whole bunch of queries, simple ones like, well, what are they for? If we imagine the brain is lit up with activity, we see looking at fMRI scanner, we can see big chunks of the sort of the blood oxygen demand, these big chunks of brain. But it actually turns out that in that chunk, there's only a handful of neurons really doing the work, and most of them are not sending any spikes at all. And also in the book, I speculated a bunch of ideas about why they would exist. So the simplest one being that obviously when we record these neurons in our experiments, we only record them from a half an hour, 2 hours at a time, a tiny fraction of their lifetime, and we give them really simple things to do. Either show them some pictures, or we get an animal the animal they're sitting in to do really simple tasks. In which case, we're just not exercising them in the regime that they would actually respond. That's one option. The type 2 dark neurons are kind of the sort of even more slightly difficult to understand cousins. So of the subset of neurons that are active, they divide neatly into ones that respond to the thing that's happening in the world. So they respond to the stimuli, they're responding to the movement or whatever. Those are the neurons that were analyzed in the papers.
Yeah.
And then there are the neurons that are active. You can definitely pick them up, but they don't respond to the stuff that you're doing. So those, in the book, I call them type 2 dark neurons on the grounds that, again, to the outside world, they would seem to be invisible.
Because people don't talk about them, right? They just don't fit their agenda, so they record them, they don't make sense, and then they don't talk about it.
Right. So particularly you pick up almost any sort of, open an issue of Journal of Neurophysiology from like the late 90s. In almost every paper, the opening of the results would say, so we recorded 150 neurons in the rats while they did press the lever or whatever they were doing. Of those 150, 80 were tuned to the task, and we're going to analyze them in this paper.
Yes.
You go, what about the other 70? What were they for? And so those type 2 dark neurons have been there forever. We've known about them, but they're not analyzed. And one of the big things that's happened in sort of recent, sort of particularly the last decade of systems neuroscience and computational neuroscience is the looking at neurons as a population and understanding that it doesn't really matter if the individual neuron is seemingly tuned to something or not. The population, you can read out stuff just fine. You can read out choice of going left or right. You can read out aspects of the stimulus. You can read out the task phase the animal was in, even if the individual neurons are passing no statistical test whatsoever for being tuned to this stuff. Because all that sort of might matter to the brain is the fact that there are enough neurons varying their activity on this particular occasion that you can tell the difference.
Also, like from the reading the How to Build Your Brain book by Elliot Smith, I learned that, and I think you can then run their analysis software as well. And basically encoding a variable in a number of 100 neurons is much easier than in a single neuron, right? So he has these like code examples where if you just want to encode a sine wave with 100 neurons, it's quite easy and you can reconstruct it really well. But with a single neuron, it's more complex, of course. So is that the same context that, only the legion of neurons could encode well enough? Or do you think it's just the way the brain works that we need multiple neurons?
Yeah, I think that's those are sort of there are parallel arguments for why you have many neurons. One, there are arguments to be made for the fact that you need a a certain level of representation to be able to encode the things you want to be able to encode. So it means you need many neurons. And there's the argument, for example, the mixed selectivity argument, sort of the Rigotti and the Panzeri arguments about the fact that there are parts of the brain, in their case prefrontal cortex, where you deliberately want to make your activity high dimensional because you're trying to combine things non-linearly. And if you do that in a few neurons, then the signals are really hard to decipher. But if you take these non-linear combinations and you represent them in this really high dimensional space, you spread them across many neurons, then they actually can be decoded really easily because they're in high dimensional space. You just put a plane between them, a simple flat plane, and you can just decode one from the other. So there are lots of good reasons why you want to have lots of neurons encode stuff rather than individual neurons.
Sure.
In the book, they were kind of talking about the flip side, which is the fact that if we believe the brain is a noisy thing, and we know lots of neurons both don't fire very much, and when they do fire, the spikes fail. So how do you encode things robustly against all this sort of stochasticity? And the answer is you encode it in lots of neurons, almost no matter how simple it is. So from that perspective, then it's more of a question of not of the complexity of the thing that you're encoding, but of it's not about many neurons opposed to one neuron, but whether or not there's more a question of any such thing as neural encoding at all at the single neuron level, and that actually the brain just is encoding stuff all the time at sort of population level, and it just so happens that because you have so many neurons, of course, some of them seem to respond reliably to particular features, so they get called edge detectors or they get called place cells or whatever.
Makes sense. Interesting. So another small detour in your book, you jump from retina directly to the primary visual cortex, mischievously skipping the lateral geniculate nucleus. But indeed, the first thing I learned from you actually on Twitter is that I think you posted this a while back, December 2018. You placed a PDF of your tattered copy of Wishaw's classical book chapter, The Decorticat Rat from 1990 there. Basically, you asked, what can rats do without a cortex? And you said a lot. And then there was a whole thread. And took me a while to read the original Wishaw PDF, but I learned a lot. So can you maybe talk a bit about that? Is the cortex that important even?
Right, it's a good question, yes. So coming from a basal ganglia group when I was trained, obviously they were big fans of this decorticat rat work, showing that cortex was not particularly necessary, so you can leave it all to the basal ganglia to do the hard work. So that it's one reason I pulled that out is because it's a huge body of work from the 80s and early 90s in which there has never been a systematic any kind of review written of it except for that one book chapter. Of a book that's out of print and isn't available as far as I could tell on any sort of any online system. And I was thinking about it, I think because I just read Randy Bruno's paper on where they were training mice to do I think a pole detection task with their whiskers. They lesioned off the whole of the barrel cortex and they could still do the task. It's like, okay, so I can take away the whole of the region of cortex that represents the whisker. How on earth are these rats learning this task? And the answer, of course, is they're doing all subcortically. The whisker information is going into the striatum, it's going to into the superior colliculus just as it hits up to cortex. And if all you're asking it to do is discriminate two simple things or hit a pole, then that's fine. And in the Wishaw chapter, there was a synthesis of all his and Brian Kolb and others' work on lots and lots of studies of what rats can do without their cortex. And if I remember rightly, the sort of the review basically says almost everything is fine, to the extent that they tested stuff. There's the kind of body of work that could really be done beautifully redone now, particularly when we have things like deep lab cut, so we can really quantify the behavior much more, in much more detail than their sort of observations of what's still going to be fairly gross, behavioral changes and stuff.
But maybe to name a few things, they can mate, they can raise pups, right? They can nurture pups. They walk on a treadmill, and they do a lot of things without a cortex. To state that.
Yeah, absolutely. Indeed. So I seem to remember the main things that they struggled with was appropriate sort of interactions with some conspecifics. So also some of the social stuff was occasionally inappropriately aggressive and stuff. And in particular, the thing they concentrate a lot in that book chapter was the problem they have with trimming their nails.
True.
So
I remember that too.
There's like a figure of a rat with really big overgrown nails because they can't groom the nails properly. Which is the suggestion that obviously you've got the perfectly fine gross motor control, but you've lost some fine motor control, and that's the most obvious sign of it. So, so one wonders, for example, although they could run fine on the treadmill, if you're going to look at their actual grippage, they probably will have issues with slipping. I imagine quite a bit on the treadmill and their motor cortex. But yes, yeah. So but sort of in terms of the things you need to do to survive, they don't seem to need cortex. Which, as you say, raises the question of what do you need cortex for?
For the nails, for trimming nails. I think that's. Do we think that might be something in the field of neuroscience, that we are too much cortex chauvinists, and that is not a well enough known fact? That I'm not, of course, cortex is not useless and used for a lot of things, but still, I think a lot of people really think of function of the brain being localized to this or that cortex region, and they might miss a lot. That's, that it would even work without that region, as you said with the barrel cortex. Do you think that's a problem? Or
yes. The short answer, yeah. So I've had the advantage of, as I said, I've been trained from a group that don't work in cortex at all, and then moving to working in cortex quite a lot. And obviously, we can see that there are various historical reasons why cortex is dominates thinking. So obviously, one is simply that we know from various very ancient lesion studies in humans that various regions of cortex are really important for various things. So back to Broca and Wernicke and so on.
Yeah.
So we have this view that a lot of what makes us uniquely human is centered in the cortex. So we want to understand the cortex. So that we go to the cortex of animals to understand the cortex of humans. That's a slight disparity, of course.
Sure.
And then, of course, there's simply the fact that the cortex is the easiest bit to record from. So just go for the top. Which means that then creates something of a kind of another bias in how we gather data. So obviously the fact that the sort of the visual cortex is the most researched part of the brain system of neuroscience is because of those two reasons. It's because we're humans, and we are most visually driven animals, so we understand how our visual system works, and it's the easiest bit of cortex to record from. It's just there at the back. You just put your electrode on the top. There it is. But then, as you say, that's all makes sense. Okay, great. But also that means that people tend to really put almost everything into cortex. So cortex does, it does all decision making. It does all value judgments. It does all everything, which is particularly bizarre to people who work on things like Parkinson's disease and go, pretty sure cortex doesn't do the things you said it does. Because you can just put up a bunch of disorders where cortex is absolutely fine, but the things are completely going wrong. So
and hypothalamus is important, I guess. If you lesion that, you die. That we know, right? So Anyways, I think this could maybe hopefully lead us to the last topic, which is focusing on your recent paper on the strong and weak principles of neural dimension reduction, published in this amazing new journal, Neurons, Behavior, Data Analysis and Theory. Can you briefly summarize what these two principles would be, or what you mean by that?
Yeah, sure. So, as we touched on, so in the last 2 decades, system neuroscience has been recording for more and more neurons simultaneously. So we're now up to the ability to routinely record hundreds of neurons at the same time. We've discussed examples already where we can record thousands of neurons at the same time. And back in February this year, a preprint from a group in Rockefeller announced they could record up to 1 million simultaneous neurons, individual neurons in the cortex of a mouse.
Wow.
with some extremely difficult to reproduce kits. So like a calcium imaging, of course. So a genetically expressed calcium sensor across all the neurons in cortex, and lots of clever tricks to put the planes of light in all the way, scanning it as fast as possible across multiple planes, lots of different lasers going off. Just kind of a tech demonstration to go, it is possible to do this.
Yeah.
Obviously, that raises the question then, of course, why would you want to do that? Scientific questions that can follow. But so obviously, the more and more neurons that we get, the higher and higher dimensional our data sets become. So even with 1,000 neurons, you've got 1,000 time series of neurons responding to things in the world. It's 1,000 dimensional data set just in the number of neurons. So the first thing that we reach for then to understand this is dimension reduction of some form. And the weak and strong principles is the question of, when we reach for dimension reduction, is it that because we think it's just a tool? So the weak principle is that it's just a tool. It's just a thing we use to make sense of the data. That helps us just grasp what it is there.
To make the data more handleable, right? As a research tool. Yeah.
So we can visualize it, so we can analyze it in this low dimensional space and make sense of it. And that perhaps that works because we're only recording these neurons over a little short period of their lifespan, so they're responding in a low dimensional kind of way for the particular task that we're giving it. The strong principle is the view that this dimensional reduction applied to these neurons is not just a tool, it's actually showing us the level at which the brain operates. Yeah. The brain is actually operating as a whole series of low dimensional dynamical systems all coupled together. And what the dimensional reduction is showing us is that low dimensional signal. That is the thing that's doing the encoding, the thing that's being passed from region to region, rather than the individual spikes of individual neurons. It's the level at which it's operating. So those are the two principles. And the idea was to lay out this, just to put it in sort of in writing, or going, you're making this choice implicitly whenever you're doing your paper. Is this what you mean? Is the first question right, to people to think about. Do you mean it's a tool? Or do you mean it's what you think the brain does?
Yeah.
And then to talk about in the paper sort of evidence for and sort of against the strong principle.
Yeah.
Given that the weak principle would kind of end up being the default position, right? So you can we can always believe it's a useful tool, no problem. Yeah. We can all agree on that. But is it something more? And something more needs a stronger debate.
Do you have a gut feeling? Is the strong principle true or not? Or is that like, do you want to lay out some pros and cons for us?
Yeah, so the most obvious, because most obvious pro is that that point of view makes sense of a whole lot of data that we have about how the brain works. So particularly in motor regions, that we can do these low dimensional projections of activity, and from that low dimensional projection, we can read out everything we want to know about the movement that's happening. And it solves a whole bunch of problems about how about how individual neurons can encode things. All those questions just go away. How do noisy individual neurons that fail and send spikes and stuff go? Well, that's not those aren't interesting questions. It's how this whole population encodes this simple low dimensional system, which means that if individual neurons fail or don't fire properly or fire whatever too much, it doesn't matter, because they're not affecting in any meaningful way the low dimensional signal inside the population. So it explains how you can have the exact same system generate the exact same movement in different animals, where the individual spiking activity looks different because the underlying dynamics are the same. Because it's a low dimensional system. So it explains a whole bunch of stuff. One of the cons, though, is that there are systems for which, for example, low dimensional activity doesn't seem to make a lot of sense. So most obviously in the sort of early visual cortices data we have, and no doubt also in somatosensory regions and in auditory regions, in which we have compelling arguments about why instead you would have a high dimensional sparse code, because any kind of low dimensional code requires that you have correlated activity between the neurons. That is the thing that's creating that low dimensional signal. But that correlated activity is, from an information perspective, redundant. So why would you just have all these neurons sending the spikes at the same time if you could get away with having just a few neurons sending a few spikes, because the energy requirements is so much less?
Of course, yeah.
So that's the big that's one of the big cons against that point of view. Yes. So yeah.
So I think one thing exactly in that regard that I personally missed from the paper is that you focus so much on which of the two principles is true, so how the brain does that, but you seemingly ignore the where it does so, right? So for me, I have this naive thought that, the same type of information could be more expanded, more decompressed in the cortex, but then be compressed, let's say, in the SDN. And it's not the same information, but they are coupled, right? We know they work together, and the basal ganglia seem to be great at dimensionality reduction, going through the striatopallidophagal bundles, so striatum, pallidum, always getting smaller. So especially in the pallidum, it's really well researched. If you think of the Percheron work, that where even the pallidal discs are always the same size, but if you move away from cortex, the receptive field of each one is bigger, right? So you have more cortex, more striatum to the same neuron. And then you go back through the thalamus back up, and it spreads out again. So and similar way could be true for, let's say, V1 versus V4, right? As you, and I want to talk about that as well. And in the last chapter of the book, you also talk about that. So could it be that just, some areas use, let's say, the strong principle more than others, or like V1 would have less compression in it than V4 would have, or motor cortex versus SDN?
Yeah, I think so. So yeah, obviously I end that paper with by hedging my bets by pointing out that yes, see, there are reasons, because we have good reasons to believe that both can be at play, but they seem to correspond to different things, different regions of the brain have to do. That perhaps indeed there are regions of the brain that conform to the strong principle. They do use low dimensional dynamics, what they do, and there are regions of the brain that, as you say, clearly expand their input. So we have, as you say, we have great evidence that V1 dramatically expands its representation from LGN, and to do so then has a sparse code to cope with the fact that it has massively expanded it. So then that would correspond to obviously something that's not the strong principle. So therefore, dimensional reduction applied to it would be the weak principle.
Yeah.
And then the big question then becomes, then how does one talk to the other? How do we get from one to the other? And as you say, there is a whole, I didn't talk about it at all, but there is a whole interesting thing about that, the ideas of basal ganglia doing some kind of dimensional reduction, which we know intuitively because of the scaling of the size of the nuclei, right? From the rats, rats, it goes from 17 million cortical neurons to 3 million striatum to about 21,000 reticulata neurons. So many orders of magnitude, and it gets expanded out again at the other end. Yes. So a whole interesting question is completely untouched is where does the translation happen, and how do you
I want to go back to Chris Eliasmith in this context, and in his book How to Build a Brain, he explains these semantic pointers, which he argues are a fundamental principle of how cognitive architectures could be built. And the fundamental principle is that they are constructs that work at both low dimensional and high dimensional states. So I think he calls it dereferencing. So you can calculate with them on a low dimensional space, but if needed, the cognitive architecture could dereference them and expand their information. I think he has good ideas, like good examples about that, where sometimes you can make a decision fast, but then potentially you want to dereference and look at the details more to some degree. I don't know if that could map to any degree on something like that, where it's sometimes helpful to make computations in a low dimensional space, such as in the SDN, of the same data, because maybe they're just better to handle. And sometimes but you need the whole information state that's maybe more in the cortex.
So that's a really interesting idea. Yes, I'd forgotten the semantic pointer idea, because I know it's supposed to be underlying the spawn architecture we discussed earlier. But yes, I guess it's kind of related to the mixed selectivity idea that you deliberately projecting this stuff up into a higher dimensional space so that you can do stuff more easily with it. I mean, that would sound like a fun idea to play with, right? This idea that you have areas that Obviously, it makes sense that kind of that sort of gross hierarchy of the brain, where we can think about regions of the brain which are really tiny. So clearly, they have to work with some kind of compressed code, and then regions which are expanded again.
Yeah.
It'd be fun to work out how that works in terms of different cortical regions. Because obviously, we have regions to believe, as I said, low-dimensional activity in like premotor cortex, where we definitely don't have it in V1. So, is it that it's possible in premotor cortex for it to move between a purely low-dimensional and a sort of more high-dimensional thing, depending on what you're doing? One can imagine that you might want some more sort of high-dimensional control over finger movement than you do over the arm movement, for example. And I know that, so people are starting to really look at finger movement coding inside motor cortex. But
didn't you even write in the book that that's something exclusive to primates, that you have a direct cortical input to hand movements because of the finger?
That's right. I think that's, some work by Roger Lemon and others that as far as we know, that primates are the only animals that have a direct projection from the primary motor cortex to the actual spinal motor neurons that connect to the fingers, as opposed to the interneurons that then coordinate the motor neurons to produce the gross movements. So that gives them, direct control over individual muscles, muscles of the forearm. Yeah. But
maybe as the very last point, it's related to what we discussed before, and I really loved that semi-last chapter of the book. You write that the intrinsic activity, let's speak about V1 again, V1 going up to V2, V4, and prefrontal cortex and temporal lobe, where the idea would be that even if you don't specifically look at something, you have as much activity in V1. Yeah,
so I give what I hope was an interesting, a new take on the sort of the hierarchical predictive processing ideas which are most well worked out, as you say, for the visual system. So we generally think of the cortical visual systems as this hierarchy of sending processing regions, where we start with V1 with the most elementary features of the environment that lines the edges. And they, through progressive sensors, they're combined together into corners and long edges in V2, and they get combined into outlines of things in V4 and into textures by combining lots of lines together to make a texture. And then further up in IT, they get turned into objects from their outlines and the colors and stuff all put together. See how this hierarchical processing going up. But we know that from various recordings that the activity in these regions is enormous and ongoing, whether or not you're looking at something. And there has been various bits of work strongly suggesting that the activity when you're looking at basically nothing, a grey screen, or keeping your eyes closed, is, at least in animals, is very, very similar to the activity when their eyes are open, which suggests that most of the activity in looking at stuff, so most of the activity is actually not then encoding stuff that's coming in, but also is doing some ongoing processing. And the ongoing processing, the best guess is what it's doing is just simply predicting the features that are going to be there in the world that you're looking at. And the more advanced idea is that the activity, because it's hierarchy going up, is a hierarchy going down too. So what's happening is that each layer is predicting what the layer below it should be sending it. Yeah. So V2 should be predicting the elementary edges that V1 should be seeing from the fact that it's predicting these lines. V4 should be predicting the lines and the corners that V2 should be sending it, because it should be seeing the outlines of these objects, and so on up so down the hierarchy. So the idea then is that the ongoing activity in these regions, there's just these predictions, because obviously our experience of the visual world is continuous. That it makes more sense that the system simply fills in things that are different and changed or wrong than it has to fill in the whole visual world every time that our brain refreshes, or however we want to think of it. And these predictive processing accounts mean that that's really simple. You simply now, instead of thinking of this bottom-up information coming from visual cortex is this complete picture of the world, what it actually is then just the difference between what's coming down and what's coming up from the retina.
Yes.
So what's being passed forward is the thing that needs to be tweaked in the predictions. And then you just continue to go on this loop with this ongoing activity. Yeah.
Exactly. So it's a loop. It's an oscillation likely in reality, right? And so I sometimes wonder, are these arrows that we sometimes draw on these diagrams, are they even, maybe they are confusing to people new to the field, because they probably aren't. So the way I see it after reading that is really that all of these regions seem to oscillate. They basically synchronize whatever their state would be with each other, right? So if you would change, let's say, V4 with an electrode, V1 would change, and the other way around. Is that correct? That they have neural networks wired up in a way that they need to add up. Wherever you change it, it would change.
Yeah,
so the whole system.
It's the indeed, there's a sense in which it makes more sense to think of the visual system as a kind of holistic thing, which is a continuous processing unit. Nonetheless, as touched on in the book, it is true. We have quite strong evidence that there is a strong feed-forward processing path in the visual system. So, we have really recent evidence of this from two different sources. One is that when we sort of Jim DiCarlo's work, where you train deep neural networks on sort of visual object recognition tasks, and you go and look at the activity in the last layer of that deep neural network, which is going to be your IT region, and you go and record from that, and you go and record an IT, and the neural responses look more or less the same. Then you perturb the objects you're giving the neural network without retraining it, and the responses perturb in the output layer the same as they perturb in the IT when you give it to the monkeys. So you have this purely feed-forward architecture is able to be able to sort of recapitulate some of the responses. Now, clearly, those responses are to the input that's being given, so obviously the input is quite feed-forward. And then we also have the lovely recent work from the Allen Brain Institute, where they've recorded hundreds of thousands of neurons now in the visual systems of the mice from mouse V1 forward, using both calcium imaging and neural pixels probes to get the spike trains. Yeah. And in both of those cases, they find quite a nice hierarchical structure to the dynamics in the visual system when shown a thing. So they give a strong sense of this sort of feed-forward process. And one reason why that might be, of course, is that when we have this feed-forward and feedback pathways, they're actually going from different layers. So there is a clear sort of feed-forward loop going on, which is the feedback is then laid on top of. Yeah. And they are, where exactly they meet in each cortical region is a little bit less obvious. Sure. So yes, so it's good to, so in terms of our thinking about how the visual system works, we still get quite far with this sort of view of this feed-forward system. Yeah. And then to think about what information actually sent by that feed-forward system is probably then when we need to think about the feedback being added to it.
So that makes a lot of sense. But if we close our eyes and we record from V1, there shouldn't be coming in much sensible information from the eyes and LGN. So, but we would, I think, in babies, we wouldn't find much, but as you say, I think in adults, we would find a lot of information, and that could still be really proactive information from top-down sources.
Yeah, exactly. Yeah. So that is the current sort of, I guess, grand theory that marries together lots of electrophysiological recordings of what happens in V1 when you're not looking at stuff, and theories of how you do inference in your visual system. Yeah. Of that, in fact, you need this top-down signal to be able to infer the information that's in the world.
And wouldn't you think if you then, take, instead of showing pictures, you would take an electrode into IT, and that's your feed forward stream going down to V1, you could record V1 and find whatever, like something that responds to that stimulation in IT.
Yes, yeah, you should. I'm struggling to recall. I think someone pointed me to a paper that's actually started doing that recently. So yes, indeed, these feedback accounts indeed suggest that if you strongly perturb the correct layer of a top level visual cortex area, you should be able to read it out downstream. So in this case, if you manage to strongly stimulate something up in layer 2-3, then hopefully you'll be able to read it out. Yes.
Great. All right. This was really fascinating. So I think a lot of these topics to some degree converge to the basal ganglia as a compression system, action selection system, but also the streams or like these strong and weak principles that might be more true depending on the region, potentially, or even at least the dimensionality. Probably strong principle is true everywhere, but how much dimensionality is reduced might be different per region, I think. So thank you so much once more for taking the time. And I think this was really interesting for everyone.
Yeah, cool. Thank you very much.